For that very kind introduction, and thank you all for coming here. This is really a great honor for me, and I'm very, very happy to receive this award, especially knowing that there are so many hardworking and talented young scientists out there that I've been competing with. So what I'd like to talk about today is some of the work that we've been doing in my group at Göteborg University, looking at the effects of temperature on fish physiology. And some of this work has been done in this experimental system. And I'm pretty sure that some of you know what this system is already, but for the rest of you, I'll actually keep you waiting a little bit, and I'll return here in a little while. I don't think I need to spend a lot of time trying to convince you that the world is slowly getting warmer. Another thing that has become more apparent in recent years is also that the incidence and severity of transient heat waves is increasing. So organisms are exposing both chronic increases in temperature, but also sort of rapid variations in temperature on top of that. This is some recent work from Oliver et al. Showing that this is also true for marine and aquatic environments. So this is obviously a big problem, but I think that one of few good things that have come out of this is that animal ecophysiology is probably a more relevant research area than ever, actually. And it has really sparked the interest of experimental biologists like myself to try to figure out what the effects of both chronic and acute increases in temperature have on various organisms. I've been particularly interested in the effects of temperature on fish, but this is not really a new research area. This is some data from my Scandinavian colleague, August Krog. He showed already over 100 years ago in a goldfish that if you increase the temperature here, there will be an exponential increase in the oxygen consumption rate of this fish. The reason for this, as we know it today, is because the cellular ATP turnover rate increases with temperature. And obviously the most efficient way of regenerating ATP is to do it aerobically. So as the temperature increases, a fish needs to make a number of adjustments to its cardio-respiratory system. And here we have a very schematic cardio-respiratory system of a fish. And if we increase the temperature here, we can expect to see an increase in gill ventilation and an increased diffusion of oxygen from the surrounding water and into the bloodstream. There are also changes in cardiovascular function. Typically, we see an increase in cardiac output to increase the circulatory convection of oxygen. With temperature, this is usually through an increase in heart rate, but it can also be increases in cardiac stroke volume. Finally, we can expect to see increases in unloading of oxygen from the hemoglobin, or we usually refer to this as tissue oxygen extraction, when the blood reaches the tissues, and then the oxygen can diffuse into the cells and further into the mitochondria and be used for ATP regeneration. So that's sort of the basic background. But obviously there are a lot of sort of knowledge gaps and unknowns surrounding this. And one thing that we've been focusing a little bit more on in recent years is that not only the resting oxygen consumption rate increases with temperature, but there are also effects on the maximal oxygen consumption rate. Typically this increases also with temperature, but sometimes we see a reduction in maximal oxygen consumption rate at high temperature, and this leads to a reduction in the scope between resting and maximal, or what we usually refer to as the aerobic scope. So that means that there can be less oxygen available to different processes at high temperature. Now, I think that many of you know that there has been quite an intense discussion whether these curves actually look like this in wild animals in nature, and what these type of curves actually means from an ecological point of view. I think that part of that discussion can be traced to the fact that we have so far relatively limited knowledge on how thermal plasticity affects the shape of these curves. Most of these curves are constructed using relatively rapid changes in temperature in lab environments. So if we consider thermal plasticity, we usually think of thermal acclimation, which is a reversible phenotypic plastic response at the level of the individual. But you know that there can also be genetic adaptation across generations, potentially, to increasing temperature. It's fairly well known that warm acclimation typically leads to a reduction in the resting oxygen consumption rate. What we have much less knowledge about is how maximum oxygen transporting capacity is affected by acclimation to higher temperature. Is it so that it actually increases with warm acclimation to increase the aerobic scope? Well, relatively few studies have looked at that. There are also many unknowns surrounding where in the actual oxygen transport cascade there are changes. What happens to cardiovascular performance, for example? What happens to mitochondrial function, and so on? There's also another pretty basic question that we know quite little about, and that is what sets the upper limit. So when we talk about temperature, we usually call the upper critical temperature limit CT max. And this is basically the temperature where the animal loses equilibrium and flips over, and in nature it would obviously be an easy prey for predators. One idea that we've been looking at in my group is whether we can trace this upper temperature limit to cardiac failure at high temperature, and whether that cardiac failure in turn is due to insufficient oxygen supply to the heart. So I'm going to return to point 2 there in a little while, but let's start looking at point 1 first. To do that, we need to return to this system. And now you see this system here from the other side, actually. And what you have here in the far background is the nuclear power plant in Forsmark in Sweden. So this is located here a little bit north of Stockholm in the Baltic Sea. And what they do here is that they direct heated cooling water from these 2 nuclear reactors into this big enclosure. And this elevates the temperature by approximately 5 to 10 degrees relative to a reference area outside of this enclosure. So here you see a temperature profile, a yearly temperature profile for the reference area. It obviously varies on a seasonal basis. And here in the red lines you see the temperature profile for the biotest area, which also varies seasonally, but it's always sort of elevated by 5 to 10 degrees. Luckily for us fish physiologists, this enclosure is full of fish, and the species that we primarily worked on is the European perch. The perch it's a relatively large species, as you can see, and they're also really robust and sturdy and useful for all sorts of in vivo experimental approaches. Here you see a perch instrumented with a flow probe and a catheter to measure blood pressure and so forth. I'm going to refer to these biotest perch as chronically warm acclimated. There might be an element of thermal adaptation also in this population, because this enclosure has been around for almost 40 years, but we really don't know so much about that, so I like to stay a little bit conservative there. So we've looked at the thermal plasticity and oxygen consumption rate of these fish, and in this particular experiment we had 3 different experimental groups. We had perch from the reference area where the temperature was 18 degrees and they were acclimated to 18 degrees. We had biotest perch that were acclimated to 23 degrees, so 5 degrees warmer. And we then had a third group where we had reference fish that were acutely warmed to biotest temperature over 24 hours. As you can see, there was a significant increase in oxygen consumption rate, just like August Krog showed in the goldfish. But when you compared the acutely warm fish to the chronically warm biotest perch, you see that there are clear signs of thermal plasticity. So basically these fish have downregulated their resting metabolic rate in this warm environment. When we look at the maximum metabolic rate, after we stress these fish for about 5 minutes to elicit the maximum response, you see that that also increases with temperature, but there are no signs whatsoever of thermal plasticity in maximum metabolic rate. When we look at the scope then, there's actually no significant difference, but if anything, there's a tendency for a higher scope in the chronically warm fish. These changes in oxygen consumption rate were nearly identically mirrored by the heart rate response in these fish. Again, the same experimental groups, and when we acutely increased the temperature, the resting heart rate increased significantly, but when we compare to the chronically warm fish, they have actively downregulated their resting heart rate. Again, when we look at the maximum heart rate response, also an increase with temperature, but again, no signs of thermal plasticity in maximum heart rate. When we look at the scope for heart rate, a pretty interesting pattern emerges where the acute warming leads to a complete abolition of the heart rate scope, but the warm acclimation leads to complete recovery of heart rate scope in these fish. So we were obviously interested in seeing what is it that causes this reduction in resting heart rate that allow these fish to recover heart rate scope in this warm environment. One of the things that changes is that there is a reduction in the intrinsic heart rate or in the intrinsic pacemaker rate. But what is perhaps more striking is that there is a very significant, almost a doubling of the inhibitory cholinergic tone on the heart. So they actively depress the resting heart rate with an increased inhibitory autonomic tone on the heart in this warm environment. There's also a slightly elevated beta adrenergic tone on the heart. Another thing we've been looking at in this perch population is the thermal plasticity of CT max, so the upper temperature limit. We've also looked at what's called a warming tolerance. So that's basically the difference between the current environmental temperature they're living at and the difference up to CT max. So basically how much warming they can tolerate on top of what they're currently living at. Here are CT max for these fish then. So about 31 degree in the reference population, but approximately 33 degrees in the biotest population. So it's significantly elevated, but it's only elevated by 2 degrees. So that means that when we look at the warming tolerance, the difference here, it's reduced by more than half in the biotest population. So I think that the sort of overall take-home message here is that these chronically warm perch can perfectly well acclimate their cardiorespiratory function to maintain a high cardiorespiratory scope at this high temperature. What they can't really compensate so much is their upper critical temperature limit. So that means that even if they seem to be doing fine in this warm environment, they live sort of dangerously close to their upper temperature limit, which makes them very sensitive to a transient heat wave on top of that. Okay, so now I'm going to switch gears a little bit and talk about why we think that this upper temperature limit might be related to the actual oxygen supply to the heart. So in the general teleost fish, there are two main ways by which the heart receives oxygen. So here you see a teleost ventricle, obviously, and all fish have a luminal venous oxygen supply. So this is basically the oxygen that's left in the venous blood that comes back to the heart after it has passed the tissues. And really the important part here is the partial pressure of oxygen in the venous blood, because that is what actually drives the diffusion of oxygen into the myocardium. Now, in about 20% of extant teleost, especially in more active teleosts like salmonids, for example, there's also a specialized coronary arterial oxygen supply to the heart. And this comes via the coronary artery that perfuses the outer compact layer of the ventricle or the myocardium with fully oxygenated arterial blood directly from the gills, basically. Obviously, it also benefits the inner spongy myocardial layer. So, why do we think that this oxygen supply is important for thermal tolerance then? This model tries to sort of summarize this. And as we've said already, there's an increase in overall organism oxygen demand with increasing temperature, but there's also an increase in cardiac oxygen demand. Both because the temperature increases, but also because the heart needs to work harder the warmer it gets. It needs to pump more blood to supply the tissues with oxygen, so cardiac output also increases with increasing temperature. As this happens, there is typically a reduction in the venous oxygen supply to the heart, and the reason for this is mainly because the tissue oxygen extraction increases as the temperature goes up, so there's less oxygen left for the heart. Often at high temperatures, we see a reduction in cardiac output. So basically the heart fails to supply the tissues with oxygen. And the idea then that we've been trying to investigate is that this reduction in cardiac output is what subsequently leads to CT max. And the reason why, it's also the reason why cardiac output fails at this high temperature. Basically, it meets a critical PVO2 threshold here where the oxygen supply to the heart becomes insufficient, and so cardiac pumping capacity decreases. Okay, so if this model is true, then we can make a number of predictions from this. First of all, we know already that warm acclimation increases CT max in fish. And if this is mediated, or if PVO2 is important for setting CT max in fish, we should expect to see an increased PVO2 at high temperatures after warm acclimation. And there are actually several reasons to believe that this is the case. First of all, there is typically a right shift in the oxygen hemoglobin dissociation curve, and there's typically also a reduced tissue oxygen demand with warm acclimation, and both of these things would elevate PVO2. So basically, the fish would meet this critical PVO2 threshold at a higher temperature, leading to a higher thermal tolerance. From an experimental point of view, we can then make the prediction that if we make the fish hyperoxic and artificially elevate the PVO2, this should lead to an elevated CT max, and it should also improve cardiac performance at high temperatures. So this is sort of the basic model. If we then add coronaries to the equation, it becomes a little bit more complicated, and we can make a couple of more predictions. First of all, we can predict that the coronary blood flow should increase with warming to compensate for the reduced PVO2, but also to increase oxygen transport to the compact myocardium. And finally, we can make the prediction that if we make any restriction to coronary flow, this should translate into a reduced cardiac performance and reduced CT max. So we've done a couple of experiments trying to see if these predictions are true. And the first experiment I want to share with you, test prediction 1 and 2. And this is work done by former PhD student Andreas Ekström, now postdoc. And what he did in this study was that he instrumented large perch with a flow probe around the ventral aorta to measure cardiac output. He also had a catheter in the branchial artery to measure ventral aortic blood pressure. And perhaps most importantly, he implanted an oxygen optode into the sinus venosus. And this allowed us to measure the PVO2 online and actually determine at what PVO2 value cardiac function failed when we increased the temperature. Here are some of the results from that study, and again we had 3 different experimental groups. We had perch from the reference area. They were acclimated to 18 degrees in this case. We had perch from the biotest area that were acclimated to 24 degrees. We then had a 3rd experimental group where reference fish acclimated to 18 degrees were tested under hyperoxic conditions by bubbling the water with pure oxygen. So we start looking at the 2 normoxic groups here, and you can see with increasing temperature there was an expected increase in heart rate, which led to an increase in cardiac output. And when we look at the venous oxygen tension of these fish, you can see that pretty much as predicted from the model, the PVO2 remained significantly elevated in the warm acclimated biotest fish. But at some upper temperature here, you see that the heart rate actually started to fail, and that translated into a reduced cardiac output because these fish couldn't compensate with an increased stroke volume. This failure in heart rate occurred at a lower temperature in the reference fish than in the biotest fish, and the same was then true for the cardiac output response. And when we calculated the PVO2 threshold when this occurred, it turned out that it pretty much occurred at a similar PVO2 threshold around 2.6 and 4 kilopascal. So this pretty much supports the model that cardiac failure happens when the fish reaches a critical PVO2 threshold. So far so good, but when we add the hyperoxic treatment group to this picture, it sort of changes a little bit. You see here that the heart rate increases just as in the normoxic reference fish, but it then fails at pretty much the same temperature. The big difference here is that the hyperoxic fish can compensate with an increased stroke volume. So they maintain cardiac output and tissue oxygen delivery much better at the high temperature. Obviously, they also have a much higher venous oxygen tension. But how do I go back? Okay, doesn't matter. So they had a significant. Okay, so they had a significantly higher venous oxygen tension when cardiac output finally failed in these hyperoxic reference fish. When we look at the resulting CT max response, however, you see that pretty much as expected and as we saw in the previous study, the warm acclimated biotest fish had an elevated CT max by about 2 degrees, but the hyperoxic treatment clearly only increased CT max by a little bit, so it sort of ended up in between the reference and the warm acclimated biotest fish. If we look at the cardiorespiratory status at CT max, so when these fish reach CT max, it becomes even more apparent that even if venous oxygen tension is nearly twice as high, stroke volume is significantly higher, cardiac output and tissue oxygen supply is significantly higher in the hyperoxic fish, it only leads to a relatively minor increase in CT max. So I think take-home message here is that cardiac failure and oxygen supply to the heart is sort of one of many physiological functions that breaks down at high temperatures, but it's not the mechanism. There are many other things that fail, and I think that neural function is an obvious thing to look at, central nervous function. Okay. So for the rest of the talk, we'll be switching species, and we've done some experiments on trout looking at the importance of coronary oxygen supply to the heart at different temperatures. And this study was done on a relatively large adult trout. Again, it's work by Andreas Ekström and colleagues. And what we did here was that we instrumented these trout with a ventral aortic flow probe to measure cardiac output, and also a tiny flow probe on the coronary artery. So this meant that we could measure cardiac output and coronary blood flow simultaneously as we increased the temperature. So this is a tricky surgical instrumentation, so we could only take this fish from 10 degrees and up to about 18 degrees. Cardiac output and heart rate increased pretty much as expected in these fish, but the coronary blood flow response was a little bit unexpected. Well, first of all, we could calculate that coronary blood flow represented approximately 4% of total cardiac output. So 4% of what the heart sends out goes directly back to the heart to supply it with oxygen. But when we look at the coronary blood flow response, you see that it first increases with increasing temperature, but then it tends to reach some kind of plateau here, so it stops increasing. That is quite unexpected, and we're not really sure why this happens, but we think it might be due to the high heart rate, which limits coronary flow, because we know that coronary flow is primarily high during cardiac diastole. So if you decrease the diastolic period, that could lead to a reduced coronary flow. It could also reflect a relatively limited coronary vasodilatory reserve in these fish. When we separated this data set into males and females, we found something pretty interesting, I think. So the black symbols here are females, and you can see that the routine coronary flow is approximately twice as high in the female trout compared to the male trout. This is probably in part related to a greater proportion of compact myocardium in females. They had about 44% compact myocardium, whereas it was only 36% in the males. The consequence of this, when you increase the temperature, is obviously that the scope for increase was much lower in females. They could only increase coronary flow by approximately 39%, whereas the males could more than double their coronary flow. This could obviously have some important ecological implications, making female salmonids more sensitive to increasing temperature and perhaps less likely to cope well with metabolically challenging conditions during spawning migrations and so forth. So I think it would be interesting to look at this in wild salmonids also. Finally, the question is then, does this increase in coronary flow have any implications for overall thermal tolerance? And we've tried to address this question in another study on juvenile trout. So in this case, we had one group of trout that had their coronary artery surgically ligated. So we basically tied off this blood vessel to completely stop coronary blood flow to the heart. And we then had a sham-operated group. They had also ECG electrodes to measure heart rate. We then exposed them to an acute warming protocol all the way up to CT max to also determine that variable. I think this is interesting from an experimental sort of more general point of view maybe, but it's also connected to real-world problems. It's fairly well known that both wild and farmed salmonids, especially as they grow older, develop coronary arteriosclerosis. And here you see an example of this. This is old data from Saunders et al. And the question is then, does this reduce thermal tolerance? So we start looking at the heart rate response then in the sham-operated and the coronary ligated fish. And the first very obvious thing you see is that the resting heart rate is significantly elevated in the coronary ligated fish, and it remains elevated throughout the increase in temperature. So this means that the coronary ligated trout reach their maximum at the lower temperature, and heart rate also starts to fail at the lower temperature. And when we look at the CT max, you see here that the CT max is reduced by approximately 1 degree in the coronary ligated trout. So again, oxygen supply to the heart is important, but I don't think it's the only thing that determines upper temperature limits. So that's all for today, and I just want to end by thanking a number of people and organizations. First of all, I want to thank this one, my wife Karen. Without her support, none of this would have been possible to do, I think. I also want to thank this little dream team here for keeping me connected to the real world and sort of reminding me what really matters in life. I've been supported by various Swedish and international funding bodies. I also want to thank the Society for Experimental Biology and the Company of Biologists, of course. And I've also been really fortunate throughout my career to have a number of great mentors. By far the most important one, as Lynn already alluded to, is Michael Axelson at Gothenburg University, but also Christina Sundell and Tony Farrell at UBC has been really important people supporting my career. And the work on temperature physiology in fish is obviously a big collaborative effort, and a lot of students and colleagues have been involved in this work. So thank you all.